Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Mir from Delhi, Hamdard University, Delhi. Today we are going to talk on module Analysis of Solanaceous Drugs under the paper Pharmaceutical and Biological Analysis. After completing this module, the student will be able to understand following. Number 1, the definition, the types and the chemistry of solanaceous alkaloids, as well as various methods that are used for the estimation of total alkaloids in solanaceous drugs, as well as individual solanaceous, the alkaloids of the solanaceous drugs. And we will also be able to understand the analysis of individual tropine alkaloids and associated alkaloids and products. Now let's first understand what are these alkaloids. I'm sure that you know that these alkaloids are naturally organic substances which are basic in nature. They are nitrogen containing substances and mostly have nitrogen as a part of their as part of their cyclic ring. There are some of those examples where the nitrogen is part of the side chain. So accordingly they are known as true and pseudo alkaloids. So they also have another property that they are physiologically active, and they are active at a very low dose. They are weakly to strongly basic in nature. So basically, in addition to carbon, hydrogen, they contain nitrogen, which is the part of either of their rings or which is the part of their side chain. So alkaloids are produced by a variety of organisms. We know that mostly they are produced by plants. So they are also produced by fungi and as well as bacteria, and some of the animals have been also been reported to produce alkaloids. They can be purified from crude extracts from these organisms by acid-base extraction. So alkaloids have a wide range of pharmacological activities. They are widely used. Some of their uses they include, like we know this quinine, which is an anti-malarial drug, ephedrine, it is an anti-asthmatic, while as quinidine is anti-arrhythmic drug, while as we know that morphine, it is a gold standard as an analgesic drug. Now these tropine alkaloids, as we understand, so it is a broader class of alkaloids, very important class of compounds which are found in many of those families, plant families, especially in Solanaceae. It is also found in Erythroxylaceae. So these families, besides like many other families, they contain these tropine alkaloids. So tropine alkaloid is a special group of alkaloids found mainly among the members of Solanaceae. The tropine alkaloids are most well-known alkaloids because they have a structure made up of a tropic acid and a tropine unit. So essentially these alkaloids, they contain a tropine acid, a tropic acid unit and a tropine unit. So some of the major tropine alkaloids are scopolamine, atropine, hyoscyamine, besides others. So we also understand like cocaine is a tropine alkaloid, but it is present in coca, erythroxylin coca. Tropine alkaloids are the basis of over 20 these active pharmaceutical ingredients which are applied as medriatics, as antiemetics, as spasmodics, anesthetics and bronchodilators. They are found in all sorts of plants, as I told you, like Hyoscyamus niger, Atropa belladonna, Datura stramonium, Erythroxylum coca, Scopolia corneolica, and Duboisia myoperites. Duboisia myoperites is the major source of many of those tropine alkaloids, particularly solanaceous alkaloids. Chemical structures of three main tropine alkaloids: hyoscyamine, atropine, and scopolamine. These alkaloids are very significant as they are mainly found in members of Solanaceae family. So these three make the basis of chemotaxonomic group within the Solanaceae family. So they act as a very significant chemotaxonomic marker. Atropine is a racemic mixture of R and L enantiomers of hyoscyamine, while as L-hyoscyamine is active form. These tropine alkaloids are known to prevent binding of acetylcholine to its receptors, so they are anticholinergic in effect. Scopolamine acting as an antagonist at both peripheral and central muscarinic receptor sites. Tropine alkaloids are found in all parts of the plant with the highest concentration found in the roots and seeds. This slide provides you the details of alkaloidal contents in different parts of some of those solanaceous drugs, like belladonna. It is actually Atropa belladonna. The dried leaves and flowering tops are used. They contain about 0.3% of total alkaloid. The roots contain about 0.45%. The hyoscyamus, which consists of dried leaves with or without flowering tops, it also contains alkaloidal content to the range of 0.04%. Duboisia, which is actually the Duboisia myoporoides, the dried leaves, which contain about 2 to 4% of alkaloidal content. So this provides the major source of the solanaceous APIs, atropine alkaloids, which are used as API. These tropine alkaloids, which are used as APIs. Details of the chemical name, molecular formula, and molecular weight of some of those tropine alkaloids. Atropine, so its molecular formula is given. Its molecular weight is 289. Hyoscyamine, it is a dimer of atropamine. So its molecular formula is C17H23O3N. Its molecular weight is again 289. And scopolamine, it is L-scopoentropine, and its molecular formula is C17H21O4N. So it has an extra epoxide ring, and its molecular weight is 303. The analysis of solanaceous alkaloids is done by various methods. Some of them, these are spectroscopic methods. Some of those, they are immunosorbent assays, and some of them, these are chromatographic or chromatographic tandem with mass spectroscopic methods. So these tropine alkaloids, there have been reports of their analysis by methods like atomic emission spectroscopy, atomic absorption spectroscopy. UV spectroscopy is a very common method that is used for their estimation and analysis. Gas chromatography or gas mass chromatography, gas chromatography tandem mass spectroscopy is a very important method for their estimation, besides HPLC. TLC as well as quantitative TLC, they are very important methods for the estimation of tropane alkaloids. So we also have this capillary electrophoresis or capillary electrophoresis with mass spectroscopy. This tandem mass is also used. There have been reports for the use of CEMS for the estimation of various of those tropane alkaloids or solanaceous alkaloids. Now let us consider the estimation of solanaceous alkaloids in general. As we know, that considerable amount of literature is available on the determination of solanaceous alkaloids. Most of these methods suggested are the variations of the general process of extraction of alkaloids. So if you understand the extraction of alkaloids, they are usually extracted, they are released from their salt form, and they are extracted with organic solvents, and then from the organic solvents they are partitioned with aqueous acidified aqueous solution, and from the aqueous acid they are released and again taken in an organic solvent. So let us discuss their details one by one in more elaborate way. First, the extraction, the ether or ether alcohol or ether chloroform mixtures are used for the extraction by the process like percolation or continuous extraction processes. So there are also like other choices, like benzene is also recommended for the extraction of these alkalides. It is more efficient, but because of its this carcinogenic effect, so nowadays it is not preferred one. The preliminary treatment of the drug with menstruum, or the solvent containing alcohol with which ultimately leads to the result in the which ultimately leads to the formation of these, or the extraction of chlorophyll, resinous matter, water soluble amines. So it also results in the extraction of saponins. So preliminary treatment, it prevents that one. Then the extraction is carried out by using chloroform after liberation of alkalides from their salts. This is done by this liberation is done by ammonium hydroxide, and then it is extracted with chloroform or these ethers, petroleum ether. That then follows the process of purification. The purification is generally carried out by alternative extraction between chloroform and dilute sulfuric acid. The alkalides are generally quite stable to hydrolysis in dilute acid solution and in neutral or slightly basic solution for the time required, that is required for our analysis. The free bases are relatively stable in solution of organic liquids. The time during which the alkalides are permitted to remain in contact with alkali must be reduced to a minimum. The alkalides, they are present in chloroform extract, are taken into an aqueous solution by salt formation, by adding by partitioning with acidified aqueous layer. After this, the impurities, they are left in the organic solvent, they are left in the aqueous layer, while as the alkalides, they are in the organic solvent. It is followed by evaporation of these organic layer. In this step, the final organic extract is evaporated to dryness to remove the ammonia and heated for a period till any volatile amines they get evolved, which have come through the purification process. It has been shown that evaporation of alkaloid residues from alcohol and heated at 100 degrees Celsius for approximately 5 hours produces no deterioration, resulting in loss of basicity or lowering of the recovery on titration. In the analysis of these tropane alkalides, sometimes the preliminary detection tests are performed. So these alkalides, tropane alkalides, they give positive results for general chemical tests for alkalides like drag and drops, Mayer's, Wegener's, and Hager's test. And some of those specific tests for tropane alkalides, they are also utilized for their detection and analysis. We understand that these alkalides, they give colored precipitates with metals and metalloids. So this is the reason for color precipitates appearing in tests like Dragondoff's, we have reddish brown precipitate. Mayer's, we have cream colored precipitate. Wegener's, we have brown precipitate, while as in Hager's we get an yellow colored precipitate. So they provide positive tests for all these general tests for alkalides. One specific test that is used for the colorimetric estimation of tropane alkalides is Vitalymic reaction. So here the residual extract, which is separated from the plants or the plant matrix, is dissolved in few drops of concentrated nitric acid and it is evaporated. Then on this dried residue, few drops of ethanolic potassium hydroxide is added. A violet to bright purple color is obtained. This confirms the presence of tropane alkalides. This reaction is the basis of colorimetric estimation of these alkalides also. The determination of alkaloidal contents by volumetric titration involves weighing of crude drug about 3 grams in a porcelain mortar. We crush it with 2 ml of ammonium hydroxide and 8 ml of water. It is poured in a flask. It is shaken with chloroform 20 ml for about 20 minutes in an ultrasonic bath. The solution is filtered through cotton. The remaining 20 ml of chloroform is added and it is again filtered. Now the chloroform solution is shaken with 2 times 10 ml of 2% H2SO4. The acid solution is made alkaline with concentrated ammonium hydroxide, and it is then shaken with 20 ml of chloroform 3 times. Then it is filtered through a cotton wool which is covered with sodium sulfate. This is done to remove the traces of water. The residue is evaporated to dryness. The alkaloidal content of the purified extract is determined by volumetric titration. So the details are as follows. The first is acid-base volumetric titration. The residue that is purified in the previous step is dissolved in H2SO4 solution in presence of 2 to 3 drops of methyl red as indicator. The excess of sulfuric acid is measured with 0.02 normal NaOH. It is back titrated with this. The content of alkaloid is then calculated as per the formula given on the screen. While as the volumetric titration involving the non-aqueous medium using perchloric acid. So here the residue of the drug is dissolved in 5 ml of chloroform and 5 ml of acetic acid is added. Then it is titrated with 0.02 normal perchloric acid in presence of 2 drops of gentiana wild as indicated. The calculation of these alkaloidal content goes as per the formula given in the lower panel. TLC can also be used for the determination of tropane alkaloids. Here the sample is prepared initially. It is separated from the plant matrix. Then that solution is applied on either a silica gel plate or Kieselguhr plate. So a mixture of chloroform, acetone, ammonium hydroxide is used as a mobile phase. So the solvent is allowed to run to about 2/3 of the overall length of this plate. So after that it is removed from the developing chamber. The solvent front is marked and the solvent is allowed to evaporate. The spots of the plate are located by spraying with drag and drop reagent. With a solvent system of chloroform, acetone and ammonium hydroxide 100:18:2, the RF values for atropine and scopolamine are 0.4 and 0.8 respectively. The RF values of the principal spot, these atropine and scopolamine, they vary as with the different mobile phases and chromatographic conditions. HPLC have also been used extensively for the analysis of solanaceous alkaloids. Several procedures using liquid chromatography coupled with UV visible or PDA detectors have been reported for the estimation of tropane alkaloids overall or individual alkaloids have been in plant matrix or in biological fluids. So these need preliminary cleanup, which is provided by liquid extraction or solid fluid extraction using cartridges. The LC is considered a reliable and sensitive method for identification confirmation of atropine, scopolamine, and other alkaloids, these solanaceous alkaloids in biological samples, fluids, and plant specimens. The limit of detection for the method for HPLC methods with a PDA detector is usually less than 1 gram per ml, so it is reasonably sensitive method. Several procedures employing liquid chromatography coupled with electrospray tandem mass have also been used and reported, and they have the beauty of estimating the atropine, scopolamine simultaneously in biological fluids. The limit of detection for LCMS method is much more accurate, and it ranges to the 10 picograms per ml for atropine and 100 picograms for scopolamine. For HPLC condition, we need a HPLC system which has a C18 column and binary or a quaternary pressure system, pump system is needed to pump this fluids, mobile phase. Mobile phase consists of acetonitrile phosphate buffer, usually at a flow rate of 1 ml per minute, and detection is usually carried out at 250 nanometers. The procedure goes like this. The specimen is poured into an activated cartridge for preliminary cleanup. The cartridge is washed with 5% methanol. The target compounds are eluted. The elute is then evaporated to dryness. The residue is then dissolved in mobile phase, and the alkylates are injected onto the HPLC system. So it is further followed by the detection of UV or PDA detection of these samples. Next, some of those hyphenated techniques have been used for the analysis of solanaceous alkaloids as so GCMs, LCMS, and CEMS, capillary electrophoresis coupled mass. So these are some of those important hyphenated techniques used for analysis of solanaceous alkaloids. Let's first discuss GCMs. So it is a system which has a fused silica capillary column and a gas chromatographic system coupled with a mass spectrophotometer. So column temperatures and injection and detected temperatures, they are varied as per the sample requirement. So the mass is set at for monitoring of ions. For example, for hyoscyamin it is 261, for scopolamine it is 375, and atropine 364. So this makes it, this is particularly for their seleniated derivatives. Next. This diagram provides you an outline of a typical GCMS. We find a GCMS system, so which consists of a long capillary column. So it acts as an separation medium. So it also acts as a ion source for the mass analyzer, which is in tandem with for detection. So the specimen, it is injected, it gets resolved into spots that are analyzed and detected, and the results are recorded. So at the same time, these elements, they are detected by a mass spectrophotometer. Next, there is a procedure for the preparation of samples for GC and its recording and the recording of these results. So this sample, about 0.5 ml of volume, it is treated with internal standards, and it is mixed with 1 ml of borate solution. So in order to render the constituents of these tropane alkaloids volatile, they are made into the derivatives, which is provided by BSTF and TMCS mixture. So they are warmed with this mixture, and this causes the derivatization of these esters. They are volatile and they are detected by they are eluted by the GC system, and they are detected by GCMS system. Next, this diagram provides an image of an LCMS system. Here we find that it is equipped with an autosampler for the injection for injecting these samples onto a column. So the column, after the column, it splits it into a UV detector and a collector. So the samples get detected, which trigger the collection of fractions. At the same time, the samples go into the mass spectrophotometer, which are detected where their mass patterns are recorded. Next, the procedure goes like this. The specimen is poured into an activated cartridge for preliminary cleanup. The cartridge is washed with 5% methanol. The target compounds are eluted. The elute is then evaporated to dryness. The residue is then dissolved in mobile phase, and the aliquots are injected onto the HPLC system. So it is further followed by the detection of UV or PDA detection of these samples. Some of those hyphenated techniques have been used for the analysis of solanaceous alkaloids as so GCMs, LCMS and CEMS, capillary electrophoresis coupled mass. So these are some of those important hyphenated techniques used for analysis of solanaceous alkaloids. Let's first discuss GCMs. So it is a system which has a fused silica capillary column, a gas chromatographic system coupled with a mass spectrophotometer. So column temperatures, injection and detected temperatures, they are varied as per the sample requirement. So mass is set at for monitoring of ions. For example, for hyoscyamin it is 261, for scopolamine it is 375, and atropine 364. So this makes it, this is particularly for their seleniated derivatives. This diagram provides you an outline of a typical GCMS. We find a GCMS system, so which consists of a long capillary column. So it acts as an separation medium. So it also acts as a ion source for the mass analyzer, which is in tandem with for detection. So the specimen is injected, it gets resolved into spots that are analyzed and detected, and the results are recorded. So at the same time, these elements, they are detected by a mass spectrophotometer. There is a procedure for the preparation of samples for GC and its the recording of these results. So this sample, about 0.5 ml of volume, it is treated with internal standards, and it is mixed with 1 ml of borate solution. So in order to render the constituents of these tropane alkaloids volatile, they are made into the derivatives, which is provided by BSTF and TMCS mixture. So they are warmed with this mixture, and this derivatization of these esters, they are volatile and they are detected by they are eluted by the GC system, and they are detected by GCMS system. As discussed earlier, that HPLC is one of the important analysis methods. So we understand that HPLC is also very important with respect to the analysis of atropine in biological samples, particularly LCMS. Atropine may be assayed accurately in plasma using radioimmunoassay techniques, while as atropine can also be assayed by various other techniques like HPLCMS, as well as GCMS, or even C, which is known as capillary electrophoresis mass spectrophotometry instruments. So LCMS instrument and the conditions that are followed for the estimation of atropine include an LCMS system consisting of following these attachments: a liquid chromatographic system, a micro vacuum degasser, system controller, and mass detector, an injection port, and a pressure system, as well as a software that handles the data. This diagram provides an image of the LCMS system. Here we find that it is equipped with an autosampler for the injection for injecting these samples onto a column. So the column, after the column, it splits it into a UV detector and a collector. So the samples get detected, which trigger the collection of fractions. At the same time, the samples go into the mass spectrophotometer, which are where their mass patterns are recorded. The CEMS is also, which is capillary electrophoresis mass spectrometer, is used for the analysis of many of those tropane alkaloids. So we need a CE system coupled with a mass spectrometer. So the electrophoretic medium consists of a mixture of trimethyl beta-dextrocyclodextrin and formic acid. So column temperature is maintained somewhere around 20 degrees Celsius, and drying gas nitrogen is used. The procedure goes like this: a specimen for the analysis is crushed using a mortar. The resulting powder and 1 ml of methanol containing 1% ammonia are placed in a test tube. The mixture is sonicated. The supernatant solution is passed through a membrane filter, and the filtrate is evaporated to dryness under stream of nitrogen. The residue is dissolved in 100 microliter of molecule water and injected on a CEMS system. Let's now discuss some of those specific estimations of tropane alkaloids. The first one is the use of GCMs for the estimation of tropane alkaloids in blood. Agilent GCMS system has been used, which has an column of fused silica capillary column. The temperature for injection and detector, they are for 250 and 280 degrees Celsius respectively. The monitoring of the ions was set as per discussed earlier, like 361, 275, 264 for their TMS derivatives. Following this method, a good linearity was obtained in the range of 10 to 5000 nanograms per ml, and the detection limit is about 5 nanograms per ml for each of the compounds. So it was applied for the determination of these alkaloids in blood after an intravenous injection. So the therapeutic concentration of atropine in blood can easily be measured using this method. This is another example where atropine and scopolamine have been detected in Datura floral nectar using LCMS system. And a Centis Express system has been used, which has a C18 column. A gradient mobile phase was used for separation of constituents. Electrospray source was operated in a positive mode. The conditions for mass spectrometer were optimized by direct infusion of the standard. So after optimizing all these column conditions and mass conditions, the method developed there was no need for a sample preparation for using for this method prior to analysis. The results show that the technique is very sensitive, reproducible and accurate. The limit of detection for using this method ranged from 75 picograms for atropine and 150 picograms for scopolamine. So a very facile method using LCMS was developed for determination of these constituents in Datura floral nectar. This is an example of use of CEMS for the estimation of atropine and scopolamine in Datura metal seeds. An Agilent systems, which has an Agilent mass spectrometer attached to it. So monitoring of ions was done at 290 and 304 M by Z. Procedure as depicted earlier, it is for these Datura metal seeds as well as for other samples that were under analysis. By this method, a good linearity could be obtained in the range of 1 to 100 micrograms per ml for aqueous solutions, and their detection limit was 0.5 micrograms per ml. So these are two official methods for the assay of atropine. In the first method, atropine is accurately weighed and then dissolved in 50 ml of glacial acetic acid. A drop of crystal violet is added. It is then titrated with 0.1 normal perchloric acid to give a green color endpoint. A blank determination is also performed for the necessary corrections. Each ml of 0.1 normal perchloric acid is equivalent to 28.94 milligrams of atropine. The other method is using TLC system. Here, the solution of atropine in methanol is prepared. Different dilutions are prepared, and then these dilutions they are applied on a TLC plate of silica So the chromatogram is developed using a mobile phase chloroform acetone diethylamine in the ratio of 5:4:1. So about the solvent front is allowed to move to about 3/4 of the length of the plate. So it is then removed, plate is removed from the chambering chamber and it is allowed to dry. The spots are located by spraying with potassium iodoplatinate solution. So colored spots, they are compared. The RF values of principal spots like atropine, scopolamine, hyoscyamin, they should correspond to their reference standards, and no secondary spot obtained from the first atropine solution should exhibit intensity equal or greater than the principal spot obtained from the second atropine solution. So the atropine solutions, they are applied in dilutions, so and quantitative as well as qualitative comparisons are made using this TLC determination, TLC method. So students, let's now summarize what we have learned in this module. First, alkaloids are naturally occurring basic nitrogenous substances having a nitrogen atom in their heterocyclic ring, and they are physiologically active basic alkaline substances. Next, propane alkaloids are the basis of over 200 APIs, among which atropine, scopolamine, and hyoscyamin form a very vital chemotaxonomic marker for the study within the Solanaceae family. So it is a very important chemotaxonomic. They make a very important chemotaxonomic marker. The two main types of propane alkaloids are atropine type and cocaine type. Propane alkaloids, they mainly contain a tropic acid unit and a tropine unit. We have also learned that most of the methods for the estimation of propane alkaloids, they are the modification of the general methods of extraction of alkaloids, which involve extraction from natural material by solvents, organic solvents particularly. It is done in presence of alkali, followed by acid extraction, and finally solvent extraction from alkaline solution. We have also understood that various estimation methods, they are either titrimetric, colorimetric, or gravimetric. Some of the instrumental methods for the estimation of solanaceous alkaloids have also been devised and reported. Many of the hyphenated techniques like LCMS, GCMs, and ECMS have been employed for the accurate and picogram scale estimation of these alkaloids in biological fluids and plant matrices. Thank you.